Hi everyone, and welcome back to my channel. I'm so glad you're here. In today's video, we are going to be covering membrane structure and function. Throughout this video, we will take a more in-depth look at the structure and function of the cell membrane that we introduced in the last chapter. We'll look at how the membrane structure leads to its semi-permeability, another concept that I had touched on in chapter 6. Finally, we'll take a look at the transport systems used by the cell membrane. These include passive transport, active transport, and bulk transport. If you like my videos, please make sure that you subscribe to my channel and turn on your notifications so that you never miss out on a new video. All right, let's get on with Chapter 7. So let's start talking about the cell membrane. From the last chapter, Chapter 6, you should remember that the cell membrane is made up of a phospholipid bilayer. A phospholipid is an amphipathic molecule. This means that it has both hydrophilic and hydrophobic regions. To review that concept, I mentioned in Chapter 6 that the head of the phospholipid is hydrophilic, meaning it is water-loving, and that the tails are hydrophobic or water-fearing. Just like these membrane lipids, most membrane proteins are also amphipathic. Because of this, the proteins can be within the phospholipid bilayer and have their hydrophilic regions protruding from it. Because of this orientation that comes about with both these molecules being amphipathic, this allows the hydrophilic regions of the proteins to be able to interact with water in the cytosol as well as in the extracellular fluid, so within the cell and outside of the cell, while their hydrophobic parts, those parts that fear water, can orient with the non-aqueous environment within the phospholipid bilayer. The figure you now see shows the currently accepted model of arrangement of molecules that we see within the plasma membrane. This arrangement is referred to as the fluid mosaic model. This model states that a membrane is a fluid structure with a mosaic of various proteins embedded within it. Let's take a look at membrane proteins and their functions. Even though phospholipids make up the majority of the membrane, the proteins determine most of the membrane's functions. There are two major populations of membrane proteins. They include integral proteins and peripheral proteins. Integral proteins actually go through into the hydrophobic interior of the lipid bilayer. The majority of these are referred to as transmembrane proteins. Transmembrane proteins actually go through the entire span of the membrane, whereas some integral proteins only go part way into the hydrophobic interior. Peripheral proteins do not go into the lipid bilayer at all, but rather they are bound to the surface of the membrane. They can be found bound to the outer portion facing the extracellular fluid, or they can be bound to the inner portion facing the cytosol or the inside of the cell. There are 6 major functions of membrane proteins. They include transport, enzymatic activity, signal transduction, cell-to-cell recognition, intracellular joining, and attachment to the cytoskeleton and extracellular matrix. I'm going to discuss in general what each of these major functions are so that you get a broad understanding of what each one means. But as you go on through biology and your upper level courses, you'll start to talk about these more specifically and talk about them specifically with what cells you're talking about and what the processes that those cells are doing. Transport proteins are integral proteins or transmembrane proteins. Transport proteins are involved in the movement of ions, small molecules, or macromolecules that allow things to move from one side of the cell to another. Some transport proteins allow the passive movement of substances, while others require ATP and active transport in order to move substances across. We'll talk later about the differences between passive and active transport. Membrane proteins can also be enzymes. Enzymes allow reactions to happen much faster. An enzyme has an active site. This is where the reactant is going to bind in order for the reaction to take place. In Chapter 8, we will discuss enzymes in much more detail. In signal transduction, a membrane protein can be a receptor. The receptor can then bind to a molecule that has a specific shape. Once the binding occurs, this allows a biochemical cascade to happen in a series of events, and this is known as a signaling pathway. Some cell membrane proteins are glycoproteins. Some of the glycoproteins can act as identification tags for that cell. These identification tags are then recognized by membrane proteins of other cells when they come into contact with one another. This binding of one cell's membrane proteins with the other cell's identification tag or glycoprotein is a very short-term binding. In the last chapter, we talked about various types of junctions that you may find between two adjoining cells. Remember, these included tight junctions, gap junctions, or even intermediate junctions or desmosomes. In order for the cells to connect with one another, there are proteins that are involved in these. The proteins that are involved in these are allowing for that intercellular joining. This type of binding is going to be a much longer lasting binding than what we saw previously in the cell-to-cell recognition. Some membrane proteins can be attached to the cytoskeleton of the cell. Remember, we talked about the cytoskeleton in the last chapter where there are many microfilaments that make up this cell skeleton. By these proteins attaching from the cell membrane to the cytoskeleton within the cell, this allows the cell to maintain its shape. It can also help to stabilize the location of certain membrane proteins. There are also other proteins that can help bind to the extracellular matrix or outside of the cell. This allows for coordination of extracellular and intracellular changes. Now let's take a look at how things move in and out of a cell. The first type of transport that we are going to look at is passive transport. Passive transport is the diffusion of a substance across the cell membrane that requires no energy. That means that this is a movement that does not need ATP. There are two things that fall into this category. That is diffusion and osmosis. Basically, diffusion and osmosis are the same thing, except that diffusion refers to the movement of substances, while osmosis refers to only the movement of water. However, diffusion and osmosis are both types of passive transport in that they require no energy in order to occur. They both allow the movement of substances or water from a higher concentration to a lower concentration, or we also refer to this as down the concentration gradient. If water or substances are moving from a higher concentration to a lower concentration, this is going to require no energy. Imagine if you spray a spritz or two of perfume in one corner of the room. Eventually, that is going to move so that the complete opposite end of the room would also be able to smell it as well. This is known as passive transport or diffusion. So where you sprayed that perfume initially, that was the highest concentration of that perfume, and then eventually it's going to diffuse through the room so that those molecules disperse through, and eventually everybody in the room can smell that. So it's going from higher concentration of that substance, in this case being perfume, to a lower concentration as it diffuses through. Think about why this happens. This happens because the cell wants to be in equilibrium. So everything wants to be equal, and when things are at a higher concentration on the inner or outer portion, it is naturally going to want to move to the other side. Now, with the cell, as we talk about different things within the cell, some things can move easier than other things, and some things just can't move unless they have an energy investment in there. But from a higher concentration to a lower concentration, things are going to want to naturally move from that higher concentration to that lower concentration to reach equilibrium. Let's go ahead and talk about the water balance of cells, or basically how osmosis works on water balance. And we're specifically talking about the outside of the cell versus the inside of the cell, or the extracellular portion of the cell versus the intracellular portion of the cell. Now, this is going to be a little bit different between cells without cell walls and then cells with cell walls. So let's talk first about how this water balance occurs in cells without cell walls. First, let me remind you that osmosis is the movement of water from higher concentration to a lower concentration. Also, let me explain to you what tonicity is. Tonicity is the ability of a surrounding solution, a solution that is surrounding the cell, that's going to cause the cell to either gain or lose water. There are different types of tonicity. The first type of tonicity that I'll describe is an isotonic solution. In an isotonic solution, the solute concentration that is outside of the cell is the same as that is inside of the cell. Therefore, in this type of solution, there will be no net water movement across the plasma membrane. Now, understand that there is always water movement in and out of the cell. However, in this case, since the outside and the inside of the cell concentrations are equal, there is not going to be a big movement of water in either direction, and so therefore there is no net water movement. Now let's take a look at a hypertonic solution. In a hypertonic solution, the solute concentration, or the concentration outside of the cell, is greater than that inside the cell. Because of this, the cell is going to lose water as water moves from the inside of the cell to the outside of the cell to maintain equilibrium. A couple things to understand in order to understand this concept. One, out of all the things that can move in and out of the cell, water can pass through the easiest. So when the cell is trying to maintain equilibrium with its outside environment, water can easily move in and out of the cell, while other things such as solutes need channels to open. So water is going to be one of the things that can easily move. The other thing that you should note by looking at this picture that I drew is that there is a higher concentration of water inside of the cell than there is outside of the cell. And so remember, osmosis works by the movement of water from a higher concentration of water to a lower concentration of water. So because there's a higher concentration of water inside of the cell and less solutes, and then on the outside, in that hypertonic solution, we see more solutes and less water. Water is going to move from its higher concentration inside of the cell to the outside of the cell. This is so that the cell can reach equilibrium, because the cell always wants to be in equilibrium. But because of this movement of water from the inside of the cell to the outside of the cell can shrink. And if the solution that is outside of the cell is very hypertonic, this could cause the cell to die if it shrinks too much. In a hypotonic solution, the solute concentration, or the concentration outside of the cell, is less than the concentration that is inside of the cell. In this case, the net water movement is into the cell, since there is more water outside of the cell than inside of the cell. the water moves from the outer solution into the cell, causing the cell to gain water. In this case, the cell swells. If the solution is very hypotonic to the cell itself, then too much water will move into the cell, causing the cell to lyse or burst. Now let's take a look at how tonicity affects cells with cell walls. We just talked about how tonicity affects cell without cell walls. So remember, animal cells only have a plasma membrane, whereas plant cells have both a plasma membrane and a cell wall. So cells with cell walls are going to react a little bit differently to this tonicity. Let's recap what we just talked about. In a normal animal cell or human cell that just has a plasma membrane, in an isotonic solution, which is this one in the middle, where the concentration outside of the cell is the same as inside of the cell, there's going to be no real net movement of water. Now remember, water does move in and out, but it's not going to affect anything, and it's at equilibrium here in this isotonic solution. In a hypertonic solution, that's where the solution outside of the cell has a greater concentration than inside of the cell. This is going to cause water to move out of the cell, causing the cell to shrink. In a hypotonic solution, a solution where the where the solution has a lesser concentration of solutes than inside of the cell, this is going to cause the water to flow into the cell, causing the cell to swell and lyse. However, plant cells have cell walls. So they have a cell wall that is rigid, and then they have the plasma membrane that is within the cell wall. So the plasma membrane is surrounded by the cell wall. So what happens in a plant cell in an isotonic solution, this actually causes the plant to be what is called flaccid. Plants like a little extra water. So in a plant cell, a normal plant cell would actually be hypotonic. So the plant cells like a little extra water. What happens here is that in a hypotonic solution where there's less concentration outside of the cell than inside of the cell, remember that causes water to flow into the cell. And so there is actually more water inside of the cell. But because the plant cell has a cell wall, a rigid cell wall, the plant cell does not burst or lyse. But the plants actually do like to be in a hypotonic solution, right? They like to be watered and have that water surrounding them. And this is where the plant cell actually likes to live, in a bit more of a hypotonic solution, which allows them to look like this. So in an isotonic solution, where the solution outside of the cell is the same as inside of the cell, the plant cell is actually referred to as flaccid, whereas in the hypotonic solution, where the solution outside of the cell is less concentrated than inside of the cell, so water will flow in, this is referred to as the turgid or normal solution for the cell. Remember, in a cell with just a plasma membrane, like these red blood cells above, an isotonic solution would be the normal. Now, for a plant cell in a hypertonic solution, this is where the outside solution would be more concentrated than the inside solution, and therefore water flows out. This is what is going to happen here. The plasma membrane is actually going to shrink within the cell wall, but remember, because of this nice rigid cell wall that protects it, it's not going to change the cell wall itself, but the plasma membrane, the solution within the plasma membrane would actually shrink, and this is referred to as plasmolyzed. Unfortunately, if the solution is too hypertonic, the membrane is going to pull very much away from that cell wall, and this is going to cause the plant to wilt, which is usually a lethal effect. Facilitated diffusion is still a passive process in that it requires no energy. However, in facilitated diffusion, transport proteins are necessary to speed the passive movements of molecules across the plasma membrane. In this case, the molecules are still moving from higher concentration to lower concentration or down the concentration gradient, again requiring no energy. However, transport proteins are needed. These transport proteins can include channel proteins and other carrier proteins. The channel proteins are responsible for providing corridors that allow these specific molecules or ions to cross the membrane. For instance, aquaporins facilitate the diffusion of water. When an aquaporin channel is open, this allows water to diffuse more freely from higher concentration to lower concentration. Different ion channels can help to facilitate the diffusion of ions. Again, when ion channels open, ions such as sodium or potassium, etc., can move from higher concentrations to lower concentrations without the need, without ATP. In contrast to the passive processes we just talked about, active transport uses energy to move solutes against their concentration gradient. So rather than going down the concentration gradient or from higher concentration to lower concentration, active transport is necessary to move solutes against the concentration gradient or up the concentration gradient, or we can say from lower concentration to higher concentration. This movement requires energy. Active transport is going to require this energy, which is usually in the form of ATP. It is also performed by specific proteins which are embedded in the cell membrane. One example is the sodium-potassium pump that uses ATP in order to work. The sodium-potassium pump is used to pump ions against concentration gradients. Sodium ion concentration is high outside the cell and low inside the cell, while potassium ion concentration is low outside the cell and high inside the cell. The purpose of this pump is to oscillate between the two, sodium and potassium. This pump moves sodium out of the cell and potassium back into the cell. Because this pump is pumping both potassium and sodium against their concentration gradients, it requires the use of energy in the form of ATP, and thus is an example of active transport. Finally, when talking about transport across the cell membrane, we can also talk about bulk transport. Bulk transport is going to occur by either exocytosis or endocytosis. First, we'll talk about endocytosis. In endocytosis, this is the process by which the cell can take in macromolecules by forming vesicles from the plasma membrane itself. Therefore, the cell will actually surround what it's going to take in and pull it into the cell, pinching it into the cell with part of the cell membrane. There are three types of endocytosis. One is pinocytosis. This is referred to as cellular drinking because the majority of what the cell takes in is fluid. Phagocytosis is referred to as cellular eating because the majority of what the cell would take in are particles. And finally, there is receptor-mediated endocytosis in which a ligand specifically binds to a receptor site on the cell membrane. So this is a very specific intake. When the different ligands bind to the receptors, then the cell will take that in the same process, pinching off a part of the cell membrane into the cell. Exocytosis is the opposite. In exocytosis, vesicles are going to be transported out of the cell. So the vesicles from within the cell will actually migrate to the membrane, fuse with the membrane, and then the cell will release the contents of that vesicle outside of the cell. Secretory cells make their product within the cell themselves, and they are wrapped in vesicles by the Golgi apparatus, and when they are ready to be released from the cell, they can then fuse with the cell membrane and be released by exocytosis. Neurons are an example of this because they can release their neurotransmitters by exocytosis. I hope this video helped you to better understand membrane structure and function and how things move in and out of the cell. If you have any questions, please don't hesitate to write them in the comments below. And remember to please subscribe to my channel so that you never miss out on a new video. Thank you.